Ladies and gentlemen, presenting Lab Chapters 14 and 15 in a nutshell. I know this says Chapter 10, but this is from 102, but it's the same material. So, let's go over some of the earliest hominins. When we're looking for human-like ancestors in the distant past, one of the ways we can figure out what exactly we're looking for is we take a look at the phylogeny of our sort of current living relatives, chimpanzees, bonobos, gorillas, orangutans, etc. And we see that back around 6 million or so years ago, we have a common ancestor with chimpanzees. Now, that doesn't mean we evolved from a chimpanzee. It means that both humans and chimpanzees are related to the same primate about 6 plus or minus million years ago. So in order to figure out when we're looking for ancestors on our lineage, which I don't know if you can see my cursor, but it is this line leading up to humans from the MRCA, or most recent common ancestor. Those are the species that we're interested in. So how can we figure out how to identify possible members of that lineage? We need to look for novel traits that have evolved along the line to humans since the split from our most recent ancestor with chimpanzees, right? All of the species that are along that line are known as hominins, so basically human-like creatures, okay? They need to be differentiated somehow from chimpanzees and bonobos. They need to have some traits that we have and chimpanzees and bonobos don't have. But they can't be obviously exactly like us. So what might those derived traits be? What are some traits that separate us from other great apes? Well, we're the only ones that habitually walk on 2 feet as our form of locomotion. There's differences in our dentition. So we have a parabolic dental arch, whereas the other apes tend to have like U-shaped arches or kind of longer arches. Our canines are smaller. We have thick enamel on our molars, and our molars are relatively large compared to our other teeth, for example. And we also have much larger brains, both absolutely and in relation to our body size. And we have a longer development period, which means instead of going straight from like early childhood to adulthood, we have a longer juvenile period where we're learning and growing, etc. We also have, more elaborate culture. You could say that if culture is shared and learned, which it is, chimpanzees, other apes, other animals can have some like sort of rudimentary culture, some things that are learned, but ours is definitely more florid and complex. So when we're figuring out what the most informative of those traits are, we could look at tool use, we could look at brain size, we could look at bipedalism, culture. Let's look at brain size for a second. From what we know already, brain size in hominin lineages didn't significantly increase until basically the jump from Homo habilis to Homo erectus. That's where you saw the first kind of large jump in cranial capacity, which is a proxy for brain size. So brain size is not like the earliest thing that we can look at as far as telling us that a certain species is a member of our lineage. It's one thing, but it's not the earliest thing to happen. I'm going to skip that because you don't really need that for the lab. So as far as tool use, that's kind of problematic because obviously the only things that really preserve in the fossil record are going to be those stone tools basically, tools that are not organic, tools that are not going to biodegrade, right? But chimpanzees, for example, are known to make some rudimentary tools, using that term very lightly, out of like branches that they strip little twigs off of, and they use those to fish for termite popsicles and termite mounds. So presumably some of the first tools that are lineage might have used would have been organic material, which we can't find in the record. And as the chimpanzee example illustrates, that might not be uniquely human. So also maybe not the very best thing that we can look at. The most important thing is bipedalism, specifically obligate bipedalism, which means that our bodies have evolved in form to make it most efficient to walk on 2 feet. Pardon me. So when I say obligate bipedalism, that's to distinguish it from these other kinds. Occasional bipedalism means like you can walk on 2 feet. Like shoot, even your dog can beg on 2 feet, right? If you're in my class or maybe in other classes, you've probably seen videos of other great apes walking for a short period of time on 2 legs, but they do it pretty clumsily and they look pretty funny. Habitual bipeds are those primates that have some adaptations that make bipedalism easier, but they also maintain adaptations that make them more efficient climbers than we are, for example. And obligate bipedalism means that that's your primary mode of getting around, is on 2 feet. So we are the only primate species around today that practices obligate bipedalism. So this is important for the lab. What can you look at skeletally that will tell you if something is probably a biped? One of the things is the foramen magnum, which is where the spinal cord attaches to the skull. The actual nerve bundle attaches to the brainstem through this hole called the foramen magnum. And in quadrupeds, it's located further back towards the back of the skull, right? Because that makes it easier for them to look forward when they're on all fours. When you're standing upright, you need to balance this big old heavy thing on its center of mass, right? So for bipeds, the foramen magnum is located more in the center of the skull. So that's one thing you can look at. We have less pronounced muscle attachments on the back of the skull because we don't need those muscles so much to hold our head up as much as quadrupeds do. However, we do have muscles that help us to have a more flexible head and that help us to hold our head steady while we're, for example, carrying a load. And so we have more, we have muscles as the other apes and primates do that attach to our skull right behind our ears. So this part that kind of juts out behind your ears is called the mastoid process. But for us, that's more important. These muscles, when you turn your head, you can probably see kind of this line of muscles and tendons that goes from your mastoids down to like your clavicle and your sternum, for example. So the mastoids are more pronounced in bipeds as well. Our spine also has a series of curves that aren't seen in as pronounced a way in other apes or primates. So it's like a sort of S-shaped curve. The lumbar curve, the lower one, helps to sort of support our weight. Kind of the S shape makes it more like a spring, right? Because we're carrying all our weight, vertically, right? So the lower spine has to really carry a lot of it. So the shape helps it to stay sort of shock absorptive, if that's a correct phrase. And it also helps us to stand upright and keep our heads upright. Versus more of like a C shape or a very moderately curved spine in other primates. Also, our lumbar verts get thick, right? The lower down the spine you go, the thicker the vertebrae get, and that's because they have to really hold a lot of weight, right? Further down you go, the more weight is on top of you. Our pelvis is one of the most informative parts. On the left of this picture, you see a human pelvis. On the right, you see a chimpanzee pelvis. You notice that the chimpanzee pelvis is narrower and taller, and ours is broader and shorter. There's a couple reasons for that. One is that this pelvic sort of bowl, if you will, is kind of holding up our internal organs, right? So there's a musculature under there. There's fibrous tissue and everything like that that helps to hold it. And this bowl shape is pretty convenient for that. The other thing is it makes for more stable walking, right? If you have a broader base, you're more stable, right? If you put something narrow on a table, it's easier to knock it over than something that's broader, right? I'll get back to another aspect of the pelvis, but also our lower limbs are longer compared to the rest of our body because that makes for more efficient walking, for example. The musculature has adapted because the bone structure has sort of shifted a little bit. So if you look at the top line with the human there, you notice that the butt muscle is thicker and meatier, right, and shorter. That plays a much bigger role. So what does the butt do? Well, it does a lot of things, but one of the most important for bipedalism is it stabilizes your hip joint. So when you're walking, bloop, 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 you know, most of the time you're walking, most of the time you're standing on one foot, right? And then you sort of switch off, and then you switch off, and then you switch off, right? So most of the time you're on one foot. When you have the other foot lifted in the swing phase of your walk, you know, physically speaking, your body wants to tilt that way because there's nothing supporting it. So what the gluteal muscles do is they cinch your pelvis to your femur. So they hold that part steady. That helps you maintain balance while you're walking. And part of the reason that our pelvises are flared out is so that that muscle attachment can allow the muscle to function in such a way that it holds the pelvis and the femur together. Okay, so that's one of the reasons that it's flared out like this. Another important thing is looking at the femur, which again is the thigh bone. On the left is a gorilla, on the right is a human, and you'll see that the angle at which the femur intersects with the tibia is a little bit different for humans, right? It comes in at more of an angle than with a gorilla, where it's almost vertical. And that's because it allows our femurs to be angled inward so that the part that's supporting the weight of the body is closer to the center of gravity, right? So when gorillas or other apes walk, they kind of do this walk like they pooped in their pants a little bit, right? They waddle back and forth. We don't have to do that because we don't have to shift our weight to get it over our tibiae, right? Because our legs are already angled in so that they're pretty much under the center of gravity as it is, right? So that's an important thing to look at when you're looking at the femur to tell if something's bipedal. There's also some other things that have to do with the foot. The thing I will note here is that in the other great apes, the big toe is more like a thumb, right? It's something that's more opposable. It can be grasping, and it's set apart from the other toes. But obviously for us, it's in line. The big toe is a bit shorter, but that bone is robust and it can support a lot of weight, and that's also important for walking. As far as why it evolved, we don't really know. That's the short version. It's not super important for the lab, but if you want to check out the PowerPoint, I will have it up. So I'm going to skip through this just to keep this video relatively short, okay? Okay, so when we're talking about some of our earliest hominin ancestors, we have a group of species that we call the pre-Australopithecines or pre-Australopiths, and that represents different genuses, geni, I don't know, and different species, right? The one that is the earliest candidate that we have so far as being a hominin ancestor is called Sahelanthropus chadensis. And that's dated to somewhere between 7 and 6 million years ago, which puts it pretty close to the split between our lineage and the chimpanzee lineage. What makes us think that it's a hominin ancestor is these little clues about bipedalism. So the foramen magnum is positioned in a more anterior spot, which means closer to the front, right? So it's more towards the center of the skull than for other apes. The canines are also smaller. The face is flatter than other great apes. The brain is still quite small, and it has a big brow ridge. So it has a mix of ancestral and derived traits, and these derived traits are traits that are along our lineage, especially these characters that make it look bipedal. The next one is Auroran tugenensis, which is about 6 million years old, this fossil. The thing that makes us think it might have been bipedal is, or like habitually bipedal, or getting towards that, is that there are aspects of the femur that are midway between humans and ancient apes. So maybe the femur is angled a little bit more, maybe the angle of the neck is a little bit different. But it maintains some adaptations in its fingers and arms for climbing that we don't have. So Ardipithecus is a really important genus. There were 2 species in this genus discovered in Ethiopia. Ardipithecus kadaba, which is from between 5.8 and 5.2 million years ago. It has a mix of primitive and derived dental traits. The toe bones might show some adaptations for bipedalism. And then there's Ardipithecus Ramidus, which is more well-known, more well-studied. We have more specimens, more complete specimens. Ardipithecus Ramidus, the most famous skeleton of Ardipithecus Ramidus is called Ardi, creative, right? Pretty short, only about 4 feet tall, from about 4.4 million years ago, and has a mix of primitive and derived traits again. So the big toe is divergent and grasping, right? Hallux is another term for big toe. So that seems to be more adapted for climbing. It has, again, a small brain around the size of chimpanzees. The limb proportions are similar to ancient apes. So apes of millions of years ago, not modern apes. The legs are a bit longer than modern apes. But as far as derived traits, it has a shorter and wider pelvis, so more like ours. The Foramen Magnum is centrally located in the skull. Despite what this picture looks like, the palms and fingers are a bit shorter than the other great apes that were alive millions of years ago from fossils that we found. And it does have thicker enamels on the molars, and its canines are not as sexually dimorphic as you might expect for other great apes. I'm going to go ahead and skip this again because it doesn't apply to the lab. Then we're moving on to Australopiths. This is another important grouping. Australopithecus is a genus. All the species in this genus were found in Africa. If you kind of lump all those species together, they date from as early as 4.5 million years ago to about 1 million years ago, and they're divided broadly into 2 groups. One is called the robusts. Sometimes those are put in a separate genus, not Australopithecus, but Paranthropus. Nomenclature, there's disagreements over how they should be classified, but just so you know. And then there's the gracile ones. Gracile means slimmer, lighter, more, I don't want to say fragile, but just not as big and bulky, right? So these are skulls of some of the robust Australopiths. On the left, you have Australopithecus boisei. Then you have Australopithecus robustus. Ethiopicus is in the middle, and then Australopithecus robustus on the right-hand side. You'll notice that they have these big cheekbones, and with those cheekbones comes like sort of wide zygomatic arches, flaring zygomatic arches, and they have this ridge of bone on the skull known as a sagittal crest. The gracile ones have a rounder skull, not really a sagittal crest. The cheekbones are still kind of wider than ours, but not as pronounced as the robust ones. So for the gracile Australopiths, they are similar to the chimpanzee in terms of body size and cranial shape. This group is made up of Australopithecus anamensis, Australopithecus afarensis, Australopithecus africanus. You can watch that on your own if you want. So afarensis is probably the most famous of these species. Lucy, if you've heard of Lucy, was Australopithecus afarensis from around 3.6 million years ago to 3 million years ago. And we are reasonably sure that they were obligate bipeds. So they didn't maintain so many adaptations for climbing. Their bipedalism was a lot more like ours. We even found footprints that were preserved under volcanic ash and dated to this period of Australopithecus afarensis, and those footprints show if you look at the close-up one, that the big toe was in line and it was shorter and it wasn't grasping, right? So that points towards obligate bipedalism. Lucy was found in Ethiopia. She's one of the absolute most complete fossil hominin skeletons. Fun fact, she was named after the song Lucy in the Sky with Diamonds, because when she was discovered, they were listening to that. The faces are a bit more jutting out. The word for that is prognathic than modern humans, but flatter than chimps. The molars were still fairly large. They might have had a really slight sagittal crest, but they probably had a more generalized diet than the robust ones, which I'll get into in a second. Cranial capacity is still pretty small. Australopithecus garhi, still small cranial capacities, lived in East Africa in general around 2.5 million years ago. There have been stone tools that have been found in layers dated to this time period, but it also kind of overlaps with the first species of the genus Homo, which is our genus, like Homo sapiens. So probably these stone tools are really associated with the Homo genus. I'll get to the technology here is called Juan Technology, and it's basically where you have a rock, right? And then you have another rock, and you sort of beat that first one with it, and you chip off some pieces. So that rock that you're chipping pieces off of is called the core. And in Juan Technology, you are shaping the core into something. In this case, it's just a chopper. You really are just taking off like a few bits to make one edge sharp. It's not that sophisticated, but it's a multi-purpose tool that you can use to like crack open bones if you're scavenging and like drink the marrow and stuff like that. And I guess you don't drink marrow, whatever, but eat the marrow. But they were like the Swiss Army knives of the day. The robust Australopiths, they had, thick enamel on their teeth, but they had large teeth in general. They had these big, bulking mandibles, and again, larger zygomatic arches, because your chewing muscles, they go under your cheekbone, right, under your zygomatic arch, and they attach up here to your head. So these larger zygomatic arches are thought to have been larger because there was a larger muscle going under them. And that sagittal crest is just an extra ridge of bone up here that these chewing muscles, all right, that you see here, they could go all the way up there and have more leverage. So that suggests to us that they had a diet that consisted of foods that were tougher to chew, right? Fallback foods, seeds, nuts, roots, tubers, not your first line foods, but things when times are tough. We've since kind of reevaluated and said that they probably ate those, but that might not have been their main diet, but they were evolved to be able to process that stuff well, because maybe climatically it was a crazy time, and periodically they would have to fall back on these foods. They also had more sexual dimorphism than the gracile forms. So males and females were of more significantly different sizes. So you have a few different species. You got Ethiophicus from 2.5 million years ago in East Africa. You have Boisei from 2.3 million years ago to 1.2 million years ago in East Africa. And then you have Robustus from Southern Africa, 2 million years ago to 1.5 million years ago. Still fairly small cranial capacities. So they are becoming bipedal. The Australopiths were obligate bipeds probably, but maybe not exactly identical to the Homo species. Relatively small brains, large teeth, but they do have thick enamel on their molars like us. I'm going to skip that. Okay, so I think that's all you need to know for these first two labs. I will post another video for the next lab. But if you have any questions, you can email me, and I will post this PowerPoint on our Canvas site as well. So I hope that helps, and I will be talking to you virtually soon.